This is Greg Demas. He'll announce himself.
All right, why don't we get going here? Thanks everybody coming for this big special day for Kristyn. It's wonderful to have such a great turnout, and thanks to Kristyn's family in the back there for making the trek all the way from Massachusetts. So welcome to sunny, balmy Indiana spring out here. So these things are always sort of bittersweet for me because it was just yesterday that Kristyn was in the lab, so now she's all grown up, she's getting a PhD, so this is a really exciting time. We got her from Massachusetts, where she got a degree at Stonehill College in 2011. And then she spent a couple of years in sort of the corporate world and realized that sort of academics was a much more fun place to be. And so came back to graduate school in 2013, and this is her 5th year. And so most of you, or a lot of you probably know that our lab looks at all things seasonal, photo period. And it's disappointing for me to say you will actually not see a short day regressed gonad anywhere in Kristyn's talk. Kristyn is the first student to actually work in a question that's kind of quite different, fits with the general theme of what we do, but quite different, and sort of really carved out her own niche and has a really wonderful story to tell, which you'll see shortly. And that's really kind of more of a developmental question in terms of how things like neonatal sickness affects an individual's development. And in this case, the individual is a hamster, not a human, but how it affects their reproductive fitness, how it affects their social development. So it's a really cool story. And then as she was working on this back 4 or 5 years ago, the whole microbiome phenomenon hit, and so we hopped on that bandwagon, and she got really excited about it. It really was the catalyst in our lab to sort of incorporate microbiome analyses as well, and really combine the two, sort of this neonatal infection and changes in the gut microbiome and how they might interact with one another. And I think it's said perfectly, very nicely by one of the reviewers of her last paper that she just submitted, where they said that it was really exciting to combine these two elements, and that was really novel. And I agree with the reviewer. That's a really important aspect of her work. So I won't belabor this too much. She's been really wonderful. I tell all my students when they get here that basically sort of when you finish your work, publish it. That way, when you get to the point of having to do your dissertation, life is really easy. You don't have to sit there and write everything up. Kristyn took it maybe a little more seriously than I expected. So she was the first one in the lab to basically accomplish what was pretty amazing. So integrative and comparative biology is a portion of her introduction. Then she published her first data chapter in brain behavior immunity several years ago. Data chapter 2 was in physiology of behavior a couple of years ago. Data chapter 3, another physiology behavior paper. Data chapter 4, physiological reports, just last year or just a couple months ago. And then what we just got news, and I think I held her up, but the goal was to sort of have it all out, and basically I held her up in terms of editing the last one. We got sort of provisional acceptance from brain behavior immunity for her last data chapter in this journal, literally just a couple of days ago. And then of course that wasn't enough, so then she had to take her discussion and publish that in this really nice review on hormones and behavior that came out last year. So she's done quite a lot, and she snuck in a bonus chapter with sort of her day job is doing science, and then her alternate career is basically working in pedagogy, and so she snuck another chapter in there, which is her pedagogical work, which she has another chapter that's in press or some form of, and then an additional one that she's writing up for some analysis she did for our animal behavior program with Kim Roswell's class. And so really quite an amazing amount of accomplishment over a short period of time. And so the other thing I want to emphasize is she was a really excellent mentor. So she really has a lot of students in the lab, undergrads that she took under her wing, and it took a while for me to even realize that sort of labs often have a regular lab meeting, and so she was organizing her own undergrad lab meeting, and so they met once a week. Actually, I caught on to this when I was sort of heading home at 5-ish, and then all the undergrads were coming in once a week around the same time, and it's because they were all coming in at 5 o'clock after hours for this undergraduate meeting that Kristyn had them regularly come in to discuss how their projects are going, science, things like that. And so she really did a wonderful job mentoring those students, and also summer students that come in Indiana. So that's really been a strength, really merging this sort of scholarly activity with this love and interest in pedagogy and teaching has really been strength, and really one not trading off with the other, because usually if students emphasize one or the other, they're not going to be as productive, and she didn't let one trade off, and really one synergized with the other. So anyway, I've bloviated for quite a long time here, so I'll pass the baton on to Kristyn, who's going to tell us about her work in modulation of the neuroendocrine system, gut microbiome, and social behavior. Kristyn.
Thank you, Greg. So I think I don't even have to give a talk because Greg already introduced everything that I've done. So I'll just skip ahead to the end to my thank yous. Like Greg mentioned, I'm going to be talking about my dissertation work, and I'm going to briefly touch upon each of my chapters as I go along. So what I first want to discuss is the fact that we all are aware that animals are faced with a number of different stressors throughout their lives, including things like finding a mate or finding territory, searching for food and finding that food source and keeping it, changes in daylight temperature and climate, as well as changes in the social environment. And so in order to actually properly respond to these different external stressors, animals have to integrate their internal physiology with their external environment. And they have both changes in behavior and in physiology when they're incorporating both of these two systems. And a number of different groups of scientists have done a great job at exploring some of these interactions. So for example, neuroendocrinologists have done a good job at explaining how the nervous system, including the brain, and the endocrine system, including hormones, have modulated social behavior, including a number of those listed on the left-hand or the right-hand side. And likewise, ecoimmunologists have done a good job at explaining how the external environment interacts with the immune system, including different antibodies, to modulate internal physiology, but less focus on how that influences social behavior. So what I really want to stress today is how all of these systems interact internally with the external environment to modulate social behavior. And what I'm going to focus on specifically in this case is looking at how external pathogens or external sickness mediate internal physiology and how those influence social behavior. And one new area of interest that we have been investigating, and Greg kind of gave that a little bit of an introduction, is the microbiome. So what we've, some recent studies have investigated how the microbiome, including the microbial communities both in and on the mammalian body, interact with both the nervous system, endocrine system, and immune system to modulate some aspects of behavior. However, I'm going to dig a little bit deeper into those interactions. And what I want to just clarify first is I'm going to be using two different terms to discuss the microbiome. I'm going to say microbiome and microbiota. So the microbiota are actually these cells that make up the bacteria, both in and on the body. The microbiome, on the other hand, is actually the genes that make up those cells. So I'm going to most often be saying microbiome because we study how those genes are affected by different treatments. But just know that if I say microbiota, it's actually talking about the cells themselves. So what I want to focus on sort of as my overall investigation for my dissertation was looking at how the endocrine system, the immune system, and the microbiome interact in very distinct ways to modulate social behavior at different times throughout an individual's life. And to do that, I'm going to walk you through each of my chapters, some more briefly than others, but I'll discuss each of my different studies. So the first question that I wanted to ask was how early life sickness could influence physiology, social behavior, and ultimately reproductive success. My second question was investigating how different types of stress in an adult could affect the microbiome and how that ultimately could influence social behavior. And then finally, sort of a culmination of all of those studies, my fifth study looked at how early life sickness influences microbiome sensitivity and if those different changes could actually affect behavior. So to do all of these studies, we use Siberian hamsters as a model system. Some of you might know a little bit about these animals. I'll give you just a little bit of background. So they're seasonal breeders, which means they integrate different environmental cues to modulate their physiology and behavior. And because of that, they're able to optimize the reproductive timing appropriately with those different changes. And because of this, it makes them very sensitive to a number of different environmental changes, including things like food availability, social stress, and immune challenge. And because they're very sensitive to these different environmental changes, they're a great model to use to investigate how different changes in both internal physiology and external environment can influence both physiology and behavior. And this is actually a real picture of our hamsters here. So again, my first question was really to investigate how early life sickness could influence physiology, social behavior, and reproductive success. And to do that, we have to be able to activate the immune system. So we can use something called lipopolysaccharide, or LPS, to activate the immune system. And what this is really just a part of the outer wall of a gram-negative bacteria. A little cartoon here to kind of explain that for you. So we take there's a piece of the outer wall of a gram-negative bacteria. What's cool is that it mimics the bacterial infection without actually causing a true infection. So we can use this and stimulate the immune system, but we don't see any passing on of that infection to different animals. But we still do see a true sickness response, including things like fever, weight loss, and decreased food intake. This is really just a great way to influence or to stimulate the immune system without causing a true infection in those animals. And there have been some studies before that have looked at how early life sickness could influence different types of physiology and behavior. However, most of them are incomplete. So what I mean by incomplete is that they often will only investigate physiology, for example. So here we're looking at a study done to look at how neonatal infection could influence reproductive development. So this is looking at luteinizing hormone here on the y-axis, which is just an important hormone for reproductive function. And along the x-axis, we have males and females, and the black bars are LPS, or those immune-stimulated animals, and the white are saline-treated animals. So what we see is that individuals who received LPS early in life had a decrease in the level of luteinizing hormone in both males and females. So it's possible that this level of hormone could influence reproductive function down the line, although many of these studies that look at physiology don't go on beyond physiology. So we don't know how those actually translate into either behavior or reproductive success. Similarly, other studies that do look at behavior often only look at non-social behaviors. So what I mean by this is behaviors that are done with an individual animal in an individual cage. So for example, here we're looking at a study, again, looking at how early life sickness could influence behavior. And in this case, we're looking at total distance traveled in the open field test. And this is a really common test done in a lot of different psychological studies that can test anxiety behavior, anxiety-like behavior. So what we see here is that this again is looking at males and females on the x-axis, and LPS is black, saline is white, and we see there's a decrease in the total distance traveled in the open field test here in males. So what this could suggest is if animals are not traveling in the open field, they potentially could have higher anxiety levels. And so we don't see the same pattern in females in this study. One thing that's really important to note about these studies is that they often look at behaviors that they try to translate into social behavior. So these are anxiety-like behaviors. Maybe they would see the same result in a social context, but we don't know that. We don't know if they would. So what I want to focus on is how early life sickness could influence both physiology as well as ultimate effects, and how they actually respond in a social context. So what's going to happen when we put them in a social context? Will they perform the same way? So to do that, I'm going to walk you through a timeline of my first set of studies. So the first thing that we do is we pair a male and a female together. We allow them to mate. And then once they've had pups, we can then give postnatal injections of either LPS, that immune stimulator, or saline as a control. Following injections, we then convene them, which means we just separate them from their parents. And then we perform a number of reproductive measurements, which I'm going to go through in just a second. So males and females both have their own set of reproductive measurements that we can take. The first of those is estimated testis volume, or ETV, which we can measure as a proxy for reproductive maturity. So we can actually measure the testes internally in the male by using calipers, and that can actually tell us when they reach a set point of reproductive maturity. So there's a number of studies before this that have looked at whether or not a male is reproductively mature if they have a certain estimated testis volume. So we can use those sort of set marks to determine if they're reproductively mature. And then we can compare that time point across groups. The second thing we do in males is we do what we're calling a reproductive challenge. So this is the hypothalamo-pituitary gonadal axis, or the HPG axis, or we can just call it the reproductive axis. And what this is showing us is that we can stimulate the reproductive system with this peptide called kisspeptin. It's not important that you remember what these peptides are called. It's just important that you know that we can stimulate the reproductive axis with this peptide, which we know is important for the onset of puberty, as well as the normal reproductive function. So if we stimulate the reproductive axis with kisspeptin, we should be able to measure downstream testosterone, because the testes, which is here, actually produce testosterone. So we can take a baseline measurement of testosterone in both groups. Then we stimulate the reproductive axis with kisspeptin, and then we can take a post-injection measure of testosterone. So we can see if the reproductive axis is functioning appropriately or not. And again, if you have any questions during it, feel free to ask, or you can wait till the end. Yeah.
Can you administer kisspeptin peripherally, or do you have to?
We inject it just IP injections into peritoneal. Yep, it's just basically putting into the systemic system, and it does the same thing. Yeah. Yeah. So in females, we have a slightly different set of measurements, but they do the same basic. They have the same basic idea. So we're looking at day of vaginal opening in our females. And one thing to note is that many female rodents don't have a vaginal opening until the day they become reproductively mature. So we can monitor females across a range of days and see when they have that vaginal opening. And then we can actually use that time point as a proxy for reproductive maturity. So once they have that opening, we can then actually see what stage of the estrous cycle they're in. You can think of the estrous cycle kind of like a female menstrual cycle. So we can take a sample from the vaginal canal, put it on slides, stain that sample, and then we can actually identify which stage of the cycle they're in. And this helps us to see if they're cycling normally or abnormally. So we take these looked under a microscope, and then across groups we can tell if a subset of those animals are cycling normally or if a subset of those are cycling abnormally. Once we've taken reproductive measurements, we then do a same-sex social behavioral assay in a subset of those animals. And what this is a 5-minute interaction between two animals of the same sex, one of our experimental and a novel, what we call intruder, is entered into the cage. We record that behavior for 5 minutes, and then we score behaviors like investigation, scent marking, grooming. And I have just a short clip to show you an example of what social behaviors look like in our animals and what we do in terms of scoring. So it's only about 12 seconds long, and this, you'll see a little bit of investigation. You'll see attacks, you'll see chases, and a tiny bit of grooming at the very end. I can play it a second time if you want to see those again once you watch it the first time. Tiny bit of investigation, then they're attacking. There's lots of chasing. Another attack, and there's a tiny little bit of grooming just for a moment in that male. And what we can do is we can slow down these videos to watch them more carefully and make sure that we're getting all of the behaviors the individual is performing. And in most of my studies, I look at both the intruder and the experimental animal as well. So once we've completed the same sex social behavior, we then have a different subset of animals that has a reproductive behavioral assay, and then another subset that has a reproductive success assay. And I'll walk you through both of those right now. So for reproductive behavior assay, the same type of situation is done. We pair an individual who's an experimental animal, except in this case, it's a novel individual of the opposite sex. We put them in the cage for 30 minutes, record that behavior, and then score all of those behaviors, including the same social behaviors like investigation, scent marking, grooming, and aggression, which are considered pre-copulatory behaviors, as well as all the copulatory behaviors like this mounting here in the male, ejaculations, as well as lordosis in a female, which is sort of the arching of the back in a female when they're preparing for mounting. And we also do a reproductive success assay in which we pair a male and a female together for 5 nights. We allow them the opportunity to mate, and then we separate the male on the 6th day, and we monitor the female for pregnancy and for litters. And once the female has a litter, we then can look at the number of pups born, the number of pups cannibalized, which is a very natural thing in most rodent species. They'll eat their young, so we can measure how often that's occurring. We can also look at litter mass as well. And finally, at the end of the study, we then collect organs and we can weigh those for mass. One thing that we do in this study is we also look at the uterine horn scarring. So this is actually, hope you can see the dark spots in the back. This is actually uterine horns taken from a female in one of my studies. And if you look carefully, you can see these little black dots that are along the uterine horns. So uterine horns are similar to a female uterus. This is actually where the pups are actually developing. And so what we can do here is we can look for scar, and we stain the uterine horns after we take them out, and we can identify these locations where the scars are these little black circles. And these show us where there are implantation sites. So these are where offspring are actually developing. And what we can do is we compare the number of offspring born to the number of scars that she has in her uterine horns. So we can tell if there are more implantation sites than there are pups. And what that would tell us is that it's possible that those pups that were developing were reabsorbed back into the uterine horns before they were carried to full term. So I'm going to bring this up a couple of times throughout the talk, and I'm going to hopefully help to show you some of the connections that are taking place across all of my studies. And so the first thing that we wanted to do was to figure out if LPS could influence physiology. So we looked at reproductive organs as well as immune organs, including the spleen and the liver. What we found is that body mass, food intake, and reproductive maturity did not change across groups, nor was it affected in either sex. And immune organs were also not affected by treatment. We also found that males were not affected by LPS in terms of reproductive physiology. So here we're looking at testosterone on the y-axis and groups across the x-axis. All of the open bars are pre-injection, so this is the baseline levels of testosterone, and all of the dashed bars are post-injection, meaning this is after the Kristyn challenge happened. And any letters that are the same, so these two A's mean they're not significantly different, and all of the B's are not significantly different from each other, but A and B are different from one another. So what this is showing us is that at baseline, both saline and LPS-treated males had there was no difference in the level of testosterone at baseline. And there was also no difference in the level of testosterone post-injection, although there was an increase in testosterone in both LPS and saline-treated males. And so we know their reproductive axis is functioning, but it's not functioning differently across groups. And similarly, we found there was no difference in paired testes mass, which is here on the y-axis, paired testes mass, and groups again on the x-axis. There was no difference in LPS or saline-treated males at the end of the study in terms of testes mass. What we did find, interestingly, is that females actually were affected by LPS treatment. So we found that LPS-treated females actually showed less normal cycling. So here we have percent cycling on the y-axis, and again, groups across the x-axis. And we can see there's a decrease in cycling in females who are treated with LPS as neonates. So we see this decrease in percent cycling. And we also found that ovarian mass was significantly lower in those animals that were treated with LPS as neonates. So here we have ovarian mass on the y-axis, saline versus LPS on the x-axis, and we see the ovarian mass is decreased in LPS-treated neonates and females. So I've shown you a little bit about the physiology in both sexes. Now we wanted to find out if same-sex social behavior is different, which ultimately could be important for competition for mates in a natural setting. And what we found is that there were no differences in either sex in all of the different social behaviors that we looked at. So investigation, scent marking, grooming, and aggression were not different across any groups in males or in females. However, we did find some interesting results in terms of mating behaviors. What I first want to introduce you to is what some of those pre-copulatory behaviors that are important in our species and many other rodent species. So many rodents will investigate their potential mate in order to understand if they're a good mate, if they should mate with them, and kind of get an idea of their identity before they mate with them. So here we have two examples. We have male nose-to-nose investigation and male nose-to-anogenital investigation. They really are what they sound like, but nose-to-nose investigation is investigating the head, neck, and nose area of the other individual. And then we have male nose-to-anogenital investigation, which is the male investigating the anogenital area of the female. And females do the same types of investigations. So they'll also investigate the head, neck, and nose area of a male, and they'll also investigate the anogenital area of the male as well. So we found no differences in any of the male pre-copulatory behaviors. So when a male was put with a novel female, the behaviors that he performed were not different across groups. However, in females, we found that LPS-treated females investigated males more often in this reproductive context. So we have here frequency of investigation on the y-axis, and we have treatment groups across the x-axis. What we found is that when females were paired with that male, they actually investigated the male more often than the saline-treated females did. This includes both head, neck, and as well as anogenital investigation, but these same patterns are seen when you separate the two types of investigation out as well. And what we also found is that females were more aggressive towards males in this reproductive context. So we found that the duration of attacks here on the y-axis, and then we have treatment groups across the x-axis. We see females tend to have longer duration of attacks when they're LPS treated as neonates. And the same pattern held true for the number of attacks, number of chases. So across the board, aggression was increased in females who were neonatally treated with LPS. One thing to note is that none of the actual copulatory behaviors were different. So males and females showed no difference in mounting, ejaculation, or lordosis. So they were still performing those copulatory behaviors at the same rate, but their pre-copulatory behaviors were different. So now we've found that there's some changes in physiology in females. There are some changes in mating behaviors in females. So now we really wanted to know if reproductive success would be influenced by these changes. And what we found is that there were no effects on reproductive success in males. So here we're looking at, when I have a male sign up here, all of these data collected here are actually from females who were paired with a male who was experimentally treated. So these are novel females who were paired with a male who either had LPS or saline treatment. What we found is that there was no difference in the uterine horn stars to pups ratio or the number of pups across saline and LPS treated groups. And interestingly, females also showed no reproductive success changes. So here we have the same measurements in females, and these are actually females who themselves were treated with LPS or saline. So we have, again, the uterine horn stars to pups ratio here, and saline versus LPS on the x-axis, and again, number of pups here on the y-axis. This is showing us that, and this is also just a subset of the different measurements that we took in terms of fecundity. So we looked at other measurements, and those also weren't significantly different. So we were pretty confident that there were no changes in reproductive success across sexes or across groups. Just to quickly summarize my first question, early life sickness affected female physiology, but not male physiology. It affected female opposite sex investigation and aggression, but not any of the same sex behavior. It did not affect any male behaviors, and it did not affect reproductive success. So moving on to my second question. Really what we wanted to find out was we found that early life sickness could influence different types of social behavior as well as different aspects of physiology. So we wanted to be able to figure out if the microbiome might be modulating some of these changes. And to do that, we really first had to figure out if we could manipulate the microbiome and if that would influence social behavior. So just to give you a little bit more information about the microbiome in general, I gave you a little bit of the information at the beginning, but again, this is a very diverse and host-specific environment, and it's made up of all different types of microorganisms, including commensal microorganisms, symbiotic microorganisms, as well as pathogens, and fungi and viruses, but I won't go too much into the fungi and viruses aspect of it. What I want to introduce you to now is a principal coordinates analysis. So this is taken from the Human Microbiome Project. This is a large group of scientists kind of across the world that have looked at the human microbiome. And what this is showing you is it takes into account dissimilarities between groups. So really what I want you to get out of this is that there are very distinct groups of microbial communities here. So each of these different areas of the body have a very distinct and unique microbial community associated with them. So this is the oral cavity here, the nasal cavity in the skin, the urogenital tract here, and the gastrointestinal tract here. And so you'll see these a number of times throughout the rest of my talk, and I just want you to be sure that you know that if they're clustering, it means they're similar, and if they're close to, or if they're clustering, they're similar. If they're separated, then they're dissimilar. That's really the kind of the take home of these figures. And what I'm going to focus on the most for the rest of my talk is the gastrointestinal system. So this really specific and unique microbial community that's here in the blue, I'm going to be focusing my talk on the gastrointestinal tract. One other thing to note is there have been many studies done on the microbiome. Most of those have been done in germ-free mice. So here we have what we're calling a conventional mouse or a conventional rodent, and they have microbes both in and on them, and they're born with microbes on them, and they're grown up with microbes in and on them. Then we have this germ-free mouse here. And what a germ-free mouse or a germ-free rodent has is no microbes in or on them. So they're born in a completely sterile environment, and they're grown up in that sterile environment. We can introduce new microbes into them if we wanted to, but most of the time we see a completely microbe-free animal, which is really important for us to understand how the microbiome is an important part of our physiology and behavior. So it's important to know there are differences between germ-free and conventional mice. However, what's really important to understand is that they're a very abnormal system. So they have lots of things that we wouldn't really want to have in a system to study. So for instance, germ-free mice have reduced digestive enzyme activity, they have reduced lymphoid tissue, they have reduced muscle layer thickness in their gut lining, and they have reduced resistance to infection. So although they're a good model to study what would happen if the individual didn't have microbes, they're quite abnormal to study in terms of normal physiology and behavior. So what we sought to do is use antibiotics instead to be able to manipulate the microbiome. So we can look at how an individual responds to their normal gut microbiome, and if we disrupt that, how would that influence their physiology and behavior? So our hamsters are not able to actually take many of the normal antibiotics that we can give to other rodent systems. They become very sick, so we can't use them. So we found a drug called enrofloxacin, which I'll abbreviate as AVX a number of times in the talk. And we can give this drug to our animals, and it's effective in cutting down microbial communities, but it still maintains a healthy organism. And it's a broad spectrum antibiotic, so it's not targeting a specific microbial community at all. It's very broad. It's safe and effective for a number of domesticated animals. And one thing to know is that it doesn't cross the blood-brain barrier. So this is something that we all have. It basically blocks pathogens and toxins from getting into the brain. And so by knowing that the blood-brain barrier, it doesn't cross the blood-brain barrier, we can be fairly confident that any effects that we see of the drug are not directly affecting the brain. So there might be other indirect mechanisms that are modulating any changes, but we know it's not from the drug actually getting into the brain itself and acting. So the first thing we wanted to do is see if we could actually manipulate the gut microbiome with this drug successfully in our hamsters. And what we did is we did a 7-day treatment of antibiotics in both males and females. And what we found is that this 7-day treatment of antibiotics did influence the microbiome. So here we're looking at the relative abundance of these microbial communities. So here's the proportion of bacterial phyla on the y-axis, and this just goes to 1 because it is a proportion. So this is just a stacked bar chart showing us the different types of bacteria, so the different phyla that are present in the microbiome. What I want you to focus your most attention on is this top portion here. So across the bottom, we have the different treatment groups. We had a pre-treatment group, or a pre-treatment time period. We took a sample at the end of that. We had 7 days of antibiotics. We then took a sample then, and then 7 days of recovery, and we took a sample at the end of that. And so what we found is that during that treatment period, if you look at that second bar in each of these groupings, and again, this is just in males for now. If we look at that second bar, we can see there's a decrease in the relative abundance of a number of different phyla in the gut microbiome. And there's somewhat of a recovery, but not a full recovery back to baseline of those same microbes. And the same pattern holds true in females, where we see there's a decrease in that second bar, which is the treatment period of antibiotics, see a decrease in the relative abundance of bacteria, and then a somewhat of a recovery, but not a full recovery back to baseline. And these water groups are their controls. So instead of giving saline, this is just water, and they're all given an oral dose of either antibiotics or water. So now that we know we can manipulate the microbiome with this drug, we then wanted to see if we could influence social behavior with these changes. So first we did a 7-day treatment of antibiotics. We then did a 7-day recovery period. This is just monitoring the animals, giving them time to recover from the treatment. A second dose of antibiotics or water during that second treatment period. So any animals who received antibiotics here also received antibiotics in that second treatment period. So it's a double dose of the same treatment. And then we gave them 7 days to recover from that as well. Throughout this entire period, at the end of each treatment period, we then did a same-sex social behavioral assay where we did, again, we paired the same-sex individuals for 5 minutes, recorded behavior, and scored the behavior. And we did that at the end of each of the treatment periods to look at behavior over time. We also collected fecal samples. That's how we actually look at the microbiome. I won't go into all those details right now, but we take fecal samples to collect the microbiome in our species. We also looked at body mass and food intake throughout the entire study. And at the end of the study, we then collected organs to look at gross anatomy. And what we found is that the second treatment period also influenced the microbiome. So this is just looking at the second treatment period. Again, this is the proportion of bacterial phyla in the gut microbiome in just males. We have water on the left and antibiotics on the right. And again, this is just the second treatment period. So this is just looking at that one time point. And if we again look at the top portion here, we can see there's a decrease in the relative abundance of bacteria that I'll go into more detail in just a second. And in females, the same situation was found. So when we have, again, water on the left, antibiotics on the right, we see a decrease in the relative abundance of the bacteria in that top portion of our dashed plot here. So now if we actually want to know which ones were actually changing, so there were 4 specific phyla in the gut microbiome that showed significant decreases in the relative abundance of bacteria during that second treatment period. And if we look very closely, you may not be able to see in the way back, but this is sort of a teal color. So if you look at that color here, there's sort of this teal color in both of the water treatments in males and females. And we don't see that same color in the antibiotic treated groups. If I sort of zoom into that and take a look here, this is actually the proportion of this specific phyla of Neuroendocrine on the y-axis. And we have both treatment groups here, water and antibiotic, on the x-axis. So what we can see is that during that second treatment period, there's a significant decrease in the relative abundance of these bacteria in the gut microbiome in both sexes. And specifically, in males, we see a near disappearance of those bacteria in the gut microbiome, but in females, there's a complete disappearance of that phyla in the gut microbiome during that second treatment period. So we see there's a significant change. And what's interesting is that this same pattern was shown in all 4 of those phyla in the gut microbiome. So this is just one example of a specific phyla that changed in the gut microbiome. And we also wanted to find out if the behavior was going to change. So again, back to this kind of interaction plot for you. We looked again at the gut microbiome and how it influences physiology. We were able to change the microbiome. We know there are manipulations that can influence the phyla in the gut microbiome, but we didn't see any changes in reproductive physiology, in immune organs, or in body mass or food intake. So the animals were all healthy, all consistently showing the same body mass and food intake. In terms of social behavior, we actually found some pretty cool results. So what we found is that 2 antibiotic treatments actually decreased aggression in males. So this is looking at number of attacks in males, and these are all the treatment groups across the x-axis. And so if you look at, no matter the color of the bar, if it's an open bar, it's a control-treated group, and if it's a dashed bar, it's an antibiotic-treated group. So if we look at just that first treatment period, we can see there's a drop in the number of attacks in antibiotic-treated males, but not significantly so. And they're able to recover their behavior in that recovery, that 7-day recovery period. And then in treatment period 2, we see a decrease in the number of attacks, and it's significant in that case. However, they are able to recover their behavior back to baseline during that second recovery period. So similarly, but sort of more robustly in females, we see that aggression is also decreased, but after that very first treatment period. So we see number of attacks here on the y-axis again, that we have during that first treatment period, we see a significant decrease in the number of attacks in females who are treated with antibiotics. And they're not able to recover their behavior after that 7-day recovery period and even after that second recovery period. So that first hit of antibiotics, that 7-day treatment period, decreases aggression, and it maintains that same low level of aggression throughout the entire study. And you might be thinking, well, maybe they're just sick and they're not behaving at all, and they just don't want to behave in any way. But we actually found this was very specific to aggression. So there were no changes in investigation, scent marking, or grooming in any of these treatment periods. So we know that the changes in behavior are very specific to aggression. And if I just draw your attention to that second treatment period where both males and females showed a decrease in aggression, and it was significant at that point. And if we think about how this relates to the level of bacteria that are in the gut, we actually found a positive and significant correlation between the number of attacks and the relative abundance of bacteria. So this is the case for a number of different phyla in the gut microbiome. This is just, again, an example of one that we saw a positive association with. So again, number of attacks on the x-axis, relative abundance of Neuroendocrine on the y-axis. We see this positive relationship. So as the number of attacks goes up, so too does the number of bacteria in the gut. And so this makes sense according to our data because we saw when the animals are given antibiotics, their bacteria are decreased as well as their aggressive behaviors. However, we don't really know what's mediating this response. So we know, so the gut-brain axis is really this communication between the gut and the brain. And so we don't know what's actually mediating these changes. However, there are a number of different hypotheses that could be modulating this change. One of those is sex hormones, including estrogens and testosterone. So we know they affect the regulation of both motor and sensory function in the GI tract, and so it's likely that it could be also modulating some of the behavioral responses. Or the stress axis, or the HPA axis, which we also know is important for appropriate stress response. So cortisol release, or the stress hormone release, we don't see that same response if we don't have the microbial content present. And finally, the immune system, or for example, cytokines, which are these immune modulators. And what they really do is they help to produce that sickness response that I first discussed in the beginning. So that fever, decreased food intake, decreased social interactions. So the immune system could really be an important player in this system. And because we know they show abnormalities in germ-free mice in the development of their immune system, it's likely this could be a player. So I'm just going to take you just for a moment, show you a few of the data that we actually collected in terms of looking at the immune system. So we wanted to find out if an acute sickness with LPS could change the microbial community. So we stimulated the immune system with LPS, just one injection of LPS to both males and females in adulthood. And we wanted to see if that changed both diversity and community. And what we found is that there were no differences in microbial communities after that immune stimulation. So here are the treatment groups. Here we have LPS and saline and pre and post injections. You can kind of ignore those a little bit. All I really want you to see here is this is another one of those principal coordinates analyses. So I want to show you that because there's clustering here, this shows us that they have similar communities. And so if there were any sort of separation between communities, we would know they might be different. And it's also not significantly different from one another. And this is just in males. So in terms of diversity and also the phyla and the families in the gut microbiome, there's also no difference across any of those. So in addition to just overall communities, there's no difference here, and there's no difference in the diversity of the microbiome, and there's no difference in the phyla present, or all the way down to family level. And the same thing is seen in females. We see a clustering of all the groups here, and so we see that they're not significantly different from one another. Just to quickly summarize this second question that I had, we found that antibiotics decreased the diversity and the relative abundance of bacteria in both sexes, that 2 antibiotic treatments in males and only 1 treatment in females decreased aggression but not other behaviors. The relative abundance of gut bacteria was associated with that aggressive behavior, and that acute sickness did not influence microbial community. So finally, my last question really is sort of bringing together all of the ideas that I had found throughout the first set of studies. What I wanted to find out was if early life sickness might influence the sensitivity of the microbiome, and if that ultimately could influence social behavior. And I want to just give you a little bit of information as to why this might be important. Why would we think this would be the case? So we know that the microbiome and the immune system develop in parallel during early development. So here, you don't need to pay attention to all the different examples here, but what I want you to really pay attention to is here we have the microbiome developing and the immune system. And if we take a look at this line here, this is the day of birth. So if we look here, there's a really quick sort of increase in the development of the microbiome after birth. And then we also see quite a few changes in the immune system shortly after birth. We see some fluctuations in the adolescent period after this weaning period, but there is sort of a homeostasis in adulthood. So we see kind of a balance in adulthood unless we challenge the system. So what's important is that any change to one of these systems, either the microbiome or the immune system, could really influence the other. So in order to get at this question, we did sort of a similar timeline. I promise you it's the last timeline that you'll see today. We did postnatal injections after pairing a male and a female together, allowing them to have pups. And those postnatal injections, again, were either LPS or saline, that immune stimulator, or the control. We then weaned the animals, separated them from their parents. We did a similar set of reproductive measurements in these animals as well. And here's where we gave them the second challenge, or that microbial community challenge. So we gave a pre-treatment period just to separate the animals into their groups, monitor them for 7 days. We gave them either treatment of water or antibiotic for 7 days. And we then had a 7-day recovery period again. And throughout this entire period, at the end of each of those treatment periods, we did another social behavioral assay. This is same-sex social behavior, scored all of those behaviors. And we also looked at body mass, food intake, and again, we took fecal samples. And we also, in addition to taking fecal samples during this period, we also took a fecal sample shortly after weaning to look at the adolescent microbiome. And at the end of the study, we collected organs to look at gross anatomy. So I'll bring you back to this again. The first thing we wanted to find out is if early life LPS would influence the microbiome. And what we found is that it didn't significantly influence the microbiome. So here we have Shannon diversity, which is one measure to look at how diverse the microbiome is, taking into account what bacteria are present and in what relative abundance they're present. And here we're looking at females and males across the x-axis, and this is saline-treated individuals and LPS-treated individuals. So there's no difference between sexes, which has been shown a number of times. In the adolescent period, there's no difference in the microbiome at that period. And there's also no difference across treatment groups. So LPS did not influence the diversity of the microbiome. One thing to note is that we also looked at the phyla and the families in the gut microbiome. And we found, though, the phyla, all the different the phyla that were present were not influenced by treatment, but 3 out of the 43 families in the microbiome were actually reduced. So very minor shifts in the microbiome, though no major shift in diversity or community structure. What we also found is that adult antibiotic treatment did influence the microbiome, similar to our first set of studies in this section. So we're looking again at a principal coordinates analysis, and this seems like a whole lot of treatment groups. Just ignore that for a second. So basically what I want you to know first off is that LPS had no influence on community structure in adulthood. So even though there were shifts, just minor shifts in adolescence, there were no shifts in the adult microbiome at this point. What I do want you to focus on is those animals who received antibiotic treatment. So here, those that are circled, all of these individuals are animals who were treated with antibiotics, and the ones that are separated from them have been treated with only water. So there's a clear separation in community structure, and so we know there's a difference in those communities as well. In females, similarly, even more so in females, there's a separation between those that received antibiotics and those that received water. And there's also differences in diversity as well as specific phyla and specific families in the microbiome. So now that we know the microbiome can be changed, we also wanted to see if any of these physiological measurements are different as well. And what we found is that there were no changes in immune organs or reproductive organs in terms of microbial shifts. However, like we saw in that very first set of studies, we saw that early life LPS did influence female reproductive physiology. So females who were treated with LPS, again, showed less normal estrous cycling and smaller ovaries. But again, antibiotic treatment had no change in any of these physiological measures. And additionally, body mass and food intake were not affected again by any of these treatments. So finally, in terms of same-sex social behavior, we did find some interesting results. However, in the first set of studies, we found changes in aggression. So we saw the animals who were treated with antibiotics showed decreased aggression. In this case, we didn't see the same effects here. So one thing I want to note is that these animals in this study were slightly younger. They were all adults in all of these studies across these time points. However, in this study, they were slightly younger, so they were more like young adults in this case. So what we found anecdotally is the animals who are slightly younger adults show less aggression overall. And so we found no changes in aggression, but really it was the case that none of these animals were aggressive at all. What we did find is some changes in other same-sex social behaviors. So here we're looking at only males. Females actually showed no changes at all in any of their same-sex social behaviors. And we have duration of investigation here on the y-axis and all of these different treatment groups along the x-axis. I want to just draw your attention to these two groups here. This is animals who received saline as neonates or LPS as neonates and then received antibiotic in adulthood. And these are showing both the pre-treatment, during treatment, and post-treatment, and again, pre-treatment, during treatment, and post-treatment again. And what I want to stress is that these individual males who were treated with, regardless of their neonatal treatment, if they received antibiotics in adulthood, they showed decreased social investigation. And this is again, during the treatment period, and we also saw during the post-treatment period, we saw no real recovery in their behavior at that time point. And again, females showed no changes in any of these behaviors. And even to a greater degree, we saw changes in self-grooming. So individuals who were treated with both LPS as neonates and then antibiotics in adulthood showed significantly increased self-grooming behaviors. So a little reminder of just a male, and this is an individual behavior, but he's still in a social context. And so this is looking at if they're behaving with that individual they're placed with, or if they're behaving individually. So what I want to draw your attention to again is this last set here, which is looking at LPS-treated neonates who received antibiotics as adults. And we see there's a significant increase in self-grooming, here on the y-axis, in males who got both of those treatments. We do see somewhat of a recovery in that post-treatment period, but not a full recovery back to baseline. And so what's important to note about both self-grooming and social investigation is that they're likely to be considered anxiety-like behaviors, and so it's possible that at this younger age, as young adults, they might be more influenced in terms of their anxiety-like behaviors. And if we think back to that first set of studies of the microbiome, we saw changes in aggression, which have sometimes been thought of as being depressive-like behavior. So it's possible that these two systems are sort of working together. So in summary, for the last question, early life acute LPS did not severely disrupt the adolescent microbiome or the adult microbiome in either sex. Adult antibiotics decreased both diversity and the relative abundance of gut bacteria in both sexes. All males treated with antibiotics as adults showed decreased social investigation. And males treated with LPS as neonates showed increased grooming only after antibiotics in adulthood. So now I'm just going to kind of give you a summary of all of the questions that I asked to kind of show you the different interactions that are taking place. So the first question that I asked investigated how early life immune challenges might influence both physiology and behavior in a number of different aspects. My second question investigated how the microbiome might influence physiology and social behavior. And my third question investigated how both that early life immune challenge and the secondary challenge to the gut microbiome might influence physiology and social behavior. What we don't know still is if the gut microbiome might influence mating behaviors here, or if we actually give individuals a chance to compete for a mate, let's say in a natural environment, if they would actually have the same results in terms of reproductive success. So in summary, females were affected by early life sickness, but they were able to adjust their physiology and behavior to reproduce. The disruption of the gut microbiome did have sex-specific effects on aggression, which we know is a critical behavior for survival. And finally, although males seem to be resistant to all of the changes in the first sets of studies, it might be possible that they're actually more vulnerable than females to behavioral abnormalities after they're predisposed to an immune challenge early in life. And so what's really critical to understand is that there are interactions taking place among all of these systems at various times throughout life, and that it's important to at least acknowledge all of these different interactions taking place. So even if we can't test each of these different systems in the course of one study, at least acknowledging that there are interactions taking place is really vital to understanding how social behavior is modulated. So with that, I want to thank lots of people, and thank you all for coming. I didn't expect so many people to come, so thank you. First, I want to thank my advisor, Greg, for his support my first semester and on. I think I just sort of hit the ground running when I got here, and he was totally open to that and let me sort of do whatever I thought I wanted to do, so thank you for that. My committee members, Jeff, Laura, and Troy, thank you for all of your support over the years and your encouragement and your help with all of my science. Lots of funding sources that I couldn't have done the work without having those. We have a ton of different lab members and collaborators that we work with, and I can't stress enough that all of the people that I've been involved with have been super helpful. And all of the undergrads here today, even previous undergrads who came to see my talk, in addition to helping with the science, they also made the lab a whole lot of fun, just being there and kind of hanging out with us. So thank you for that. This is actually us in animal quarters, all dressed up to go into our animal room. I also want to thank my Scholarship of Teaching and Learning friends and collaborators. So first, I want to thank Whitney Schlegel, who's here today. This is actually a picture of us in California at a conference together. And I really want to thank her for taking me under her wing and teaching me how to do all of the Scholarship of Teaching and Learning things, and also being a great friend. Katie Kearns in the Center for Innovative Teaching and Learning. She really helped sort of gather the data that I needed for my studies and has been really encouraging all the way through. Drew Koki and all of the past tutors. So I worked in a tutoring program on campus for a couple of years. And in addition to being fantastic people, they also really encouraged my scientific work as well and my Scholarship of Teaching and Learning work. So thank you for that. Drew's here today as well. Kim Rossvall for being a great collaborator, both in terms of my science as well as my teaching and learning, allowing me to do a great collaboration with her class. And 2 graduate students, actually this is Drew and the past leaders as well, this bottom picture here. 2 graduate students who previously graduated, Sarah Keesum and Erica Waters, have been really great mentors in terms of scholarship teaching and learning. They're here in this picture at a conference in Boston. I want to thank my friends, both near and far, and there are lots of other people that have been super helpful. So I hope I don't embarrass any of you with these lovely pictures that I found, but thank you guys for being super supportive and always helping me in terms of science, in terms of just daily things happening in my life. You've been great all the way through. My family who's here today, thank you for supporting me not only now, but also the last 29 years of my life. Thank you so much for all of your help along the way. I couldn't have done it without you guys. And finally, Thomas, thank you for all of your help throughout the last 5 years. Throwing me parties after each study that I completed and picking me up in the middle of the night after behavioral assays. I couldn't have done it without your love and support, so thank you. And with that, I want to open it up for questions. And I thought I might remind you of some of the systems if you have any questions.
How do they smell when you give them antibiotics? I just keep thinking about the aggression versus the investigating, and it seems to me that like maybe there's nothing.
It's a good point. So the antibiotic itself does smell funny. We actually thought they weren't going to take it because it's such a really strange smell. But we give the antibiotics with a pipette, and they like grab onto it and drink it like a baby, basically. So one thing that we didn't actually test like empirically, but we know that in males anyway, the scent glands when we're putting them in his behavioral assays, they're just covered. So all these hamsters have scent glands, and they produce all kinds of different molecules from their scent glands to both attract and keep males away. And they were super covered in all kinds of junk. And so we didn't actually look at that empirically, but it is definitely something to think about. And we even thought of just like taking samples from it, but we didn't know what we would end up doing with it. But it's definitely something that could be absolutely changing, and it would be something to kind of look for in the future. Yeah.
So first, related to that, when you give search and rescue dogs antibiotics, they like often have a period where they just like can't smell and they can't find anything. So like it does seem to affect their sensory system.
Yeah.
But then second, my thought was how does LPS as a simulation of sickness, how might that differ based on other papers to an actual infection where you get competition between microbes and that sort of thing?
That's definitely a good point. And I think there are other ways of obviously stimulating the immune system. I would assume it would be quite different because there is a lot of competition, and I definitely think in terms of that last study where we had the microbial changes, we would definitely see something different. But we were just trying to see if it's just like a small stimulation. This is really a very minor immune response. So, and they are over it very quickly, and so I don't know the answer, but I would assume it would be very different only because there's competition between those. And we know there's different forms of LPS depending on the microbial composition. So if we introduce a new bacteria that might have a different LPS, it's possible that those changes would be very different. Yeah, absolutely.
Thinking about that beautiful correlation between aggression, behavior, and I forget which phyla of bacteria. I'm completely ignorant of what you can and cannot do with the microbiome. But there's any way to, instead of knocking it out, to drive expression or abundance of that particular phyla and see if you can get, continue on with that correlation?
Right. So our next, actually, what we were hoping to do with actually one of our undergrads who was doing a study is give a fecal transplant. So take some of the, either manipulate or not manipulate the microbiome, and then give a fecal transplant to those individuals to see if this know there are some studies that have given almost like a probiotic, but it really is just giving them a specific type of bacteria. It's difficult to do because when you're manipulating the communities, you're also changing the levels of pathogens, and so keeping the animals healthy is hard, and you also have to maintain, by us keeping the same body mass, we knew that it wasn't an issue in terms of the animal becoming smaller and healthy. So by giving them another pathogen, even if it's a commensal, say, organism, is a difficult task to do, but I think there are a few studies out there sort of trying to drive up the abundance of the bacteria and see if the same changes are more robust. Yeah.
Also for those aggression studies, were those resident intruder assays, or?
Yep. Mm-hmm.
And all of the bacteria guys will be intruders or residents?
The residents. Yep.
That's two.
Yeah.
So, in the first study, you had this early life LPS and same-sex social behavior, not influence. Right? Folks love the early life LPS as like a model for autism and all sorts of impairments.
Mm-hmm.
Why do you think there are differences here? Is it like a species thing or a dose thing?
I think it's an age thing, to be honest. So what we were talking about actually recently at one of our, I think, a lab meeting, is actually doing an empirical study of having individuals of Siberian cancers at different age groups, and then looking at just purely behavior, nothing else but behavior, because we've over the years kind of like anecdotally noticed changes and noticed some things decreasing and increasing depending on the age, but we don't know if it's actually something that we should be, we basically all of our studies are adults, and so we have a set time point, and when they become an adult, they're reproductively mature, they're at this certain time point. But I think there's lots of variability between like an adult who's fairly young and then an adult who's maybe like on their way out. I think there's lots of range, but they're still reproductively active and mature, and so it's hard because they're still cycling and the males are still producing sperm, so it's a difficult thing to do. But I think trying to get some of our undergrads to do a study where they're just looking at the behavior across age groups.
Didn't NIH just say they want more aging included as a variable? I think I heard that somewhere. Right?
I don't know.
Okay, so the second question. In study number 3, where you saw effects on social investigation and self-grooming, if you had to pick a hormone that you think might be involved in the explanation of that,
Not oxytocin.
We could measure oxytocin, we could measure the cortisol. So we're actually, we have another study that an undergrad who's doing a senior thesis, or a study of hers, is looking at basically that first microbiome study, so just antibiotics and just looking at behavior, and she's measuring estradiol and cortisol over the course of those time points. So hopefully we'll know a little bit about what might possibly be driving at least the aggression with just antibiotics, but we don't know yet. But the hope is that something will come out of those to at least get a little bit of an idea of what might be driving some of those changes.
Other questions for Kristyn? Easy audience.
I know. Well, thank you.
Let's thank her.
I'm gonna say goodbye to the crowd. That's it. Over and out.